Dear student friends, I am Dr. Godbole Mahendra Tukaram, welcoming you all in our today's session, in today's presentation. The today's presentation, as you are known, this is about the cell biology and the cytology. The cell biology and cytology is supposed to be the most important or the primitive research-oriented work, we may say, in a biology. It was a pioneering work. Cella is supposed to be a Greek word. Cella means the room. The honeybees. Honeybees are supposed to be residing in a small, that is a benzene-like structured holes or its houses. Earlier, cell was invented by the Leeuwenhoek. It was just looking like that. The cella means a compartment, individual rooms. Now, later on, different kinds of the research was done by the different biologists and the different scientists, or the genetic engineering was also helped to enhance this branch. Okay, so in cell biology we will discuss about the different cell organelles. The individual cell organelles information is very important. If you may see the certain previous year question papers to qualify or to crack this National Defense Academy's examination, that is NDA's exam, so you will observe different kinds of the questions were supposed to be arised from this section. Right from the 2019, let us see the data from the 19 to 2024. There was the average, we may say, the 3 questions every year. We may say the 3 questions was arised from this area of the biology. And right, sometimes it may be, let us take an example of in 2019, there were the 5 questions was arised in the first and the second. You know that this examination is conducted twice. Okay, so this is very important area. Now the questions, those who are going to framing, those are the most intellectuals. They are the university professors, and although you are supposed to be studying about the cell biology and cytology, but the wording makes some confusion to the students. Yes, please. And therefore we are going to studying detailed about the cell biology and the cytology. There are certain scientists we are going to introducing earlier, that is Matthias Seldens and the Theodore. He has invented certain he was the zoologist, and there were combined work right from the 1838 to 1839. They were carried out certain work on the cell biology, and they were invented the cell theory. The cell theories question were also arised. Later on, another scientist, he was the Rudolf Virchow, and he has explained the omnis cellula e cellula. Omnis cellula e cellula means what? This is supposed to be connected to the cell biology, but it is somewhat different. It is somewhat different. This omnis cellula e cellula means the cells are arising from the cell. That is a cell division. For example, earlier we were the babies, we were the very small, but nowadays we are fully grown. So this is what happening with us. It is a cell division. Cell may divide 2 ways. Which are those? Mitosis and meiosis. Okay. The multiplication of the cell means the cell division, but either mitosis or meiosis. It is a mitosis, multiplication, and meiosis can take place in the reductional cell division for a sex cells, we may say, either in a spermatogenesis, or if we may talk about the plants, it may be for the process of the formation of that ovule, or the that is angiosperms, in different plants. Especially that microsporangia. Within the microsporangia, there will be a different reductional cell division, and ultimately the spore will be formed, and that spore may fuse with the female reproductive organ, that is gynosium, and ultimately, or the ovule, and ultimately either the seed or the fruit may be developed. The next point, or the coming to this reproductive phase or the reproductive concept, sometimes there are many questions were arising from this area also, from reproduction, especially in biotechnology. Those students are supposed to be going to preparing for the NEET examination. for the MBBS, either for the Indian and other abroad examination, for an international examination. That is also important, because what is how the seedless fruits can be formed. Okay, that is Ag3 acid is also important for the formation of the seedless fruits like the banana and other fruits that we will discuss while talking about the botany. Okay, so the Rudolf Virchow says that the omnis cellula means the cell is arising from the another cells, and the cell arises from a pre-existing cell. Cell is going to, either the cell may be prokaryote or cell may be the eukaryotes. There are the different kinds of the classification of the cells. What is meant by the prokaryote cells? Do you know that prokaryotes means lack of the nucleus? Prokaryotes. And eukaryotes means in the presence of the nucleus. If the nucleus is there, means the center is there. That center will have a leadership in the cell. So he may start the cell division. Okay, the DNA replications, RNA may also synthesized from the nucleolus. Within the nucleus there is a nucleolus, and within the nucleolus there will be the RNA. Okay, so that we will discuss. There are the certain fundamental things we have to study about the principles of the modern cell theory. So why we are going to talking about the cell is supposed to be the fundamental unit. When we talk about the physics, when we talk about any material science, that atom is supposed to be the structural and functional unit. Okay, likewise here in the life science, the cell is supposed to be the most important. And even nowadays in in cytology or cell biology, you know that is there are certain biotechnology is going to developing with the special reference of the medicinal biology. Okay, even single cell may be able to produce an entire organ. Okay, that is a callus structure can be developed. Likewise, the cell, it is an important and functional unit of the organism, any living organisms, and it is composed of the more cells. Sometimes the cell is in individual. For example, in unicellular organisms, that is a single cell itself is enough to lead the life. But in multicellular organism, the cells may be many. So how much cell may be exist? Let us take an example of the human body. Okay, while talking about the physiology, human physiology, and just to keep aside of those cells, let us take an example of the veins and venules or the capillaries in our body. It having a very huge network, and with the help of if you may connect all the different small veins and venules, then we will be able to tide the entire globe 2 times. That is 95,000 kilometer or 60,000 miles. Okay, network may be created by those small venules and venulets and veins. That much cells which are supposed to be existing in our body. While talking about the size of the cell, cells are too much small. Let us take an example. We are supposed to be measuring the cell in the scale of the millimicron. Okay, the prokaryote cells are supposed to be very small. Those may be ranging from the 0.1 to 5 millimicron, and the eukaryotic cells may be greater. Their size may be greater up to 100 millimicrons, and 1 millimicron means it is a part of the 1 millionth of the 1 meter. 1 millionth, one micron means a 1 million by 0.000 means जो मिलियन के पहले जो फिगर आ जाएगी that. Okay, coming to this, there are certain theories which were or the invented. So he has created, that is supposed to be the Robert Hooke. He is supposed to be the father of the cell biology. He has created first very single microscope, and he was able to observe the cell the more than the human vision से ज्यादा देखने की जो कला थी उन्होंने डेवलप की बाय मेकिंग ए कंपाउंड माइक्रोस्कोप, एंड ही मे बी एबल टू सी द ऑब्जेक्ट 1000 टाइम, okay, as compared to the human vision. While examining thin slices of the quark जो होता है, जो thin sections बोलते हैं हम उसे quark. Okay, those are very small sections, and just like under the microscope, for example, we are going to preparing any small slides. Okay, So we may create very small thin sections so that we can see properly. So thin slices of the quark under the self-designed primitive. Primitive means very earlier microscope he has been. Nowadays electronic microscopes are very modern techniques are used to create or to observe the primitive compounds microscopes, and the hook observed the networks of a that is an empty, tiny and honeycomb like structure. He has observed this type of the structure. So earlier human visions, to see such a structure, it was beyond the normal vision of the human being. But with the help of the compound microscope, he was able to see, observe this kind of the honeycomb-like structure. Okay, while talking about the cell's length also, so the cell length is also very long. So for example, which are the longest cell in our body? Neuron cells are supposed to be as long as 1 meter. And while talking about the broad or the size of the cell, which is supposed to be the larger cell in our body, larger cells. Okay, talk about any cell in the world which is supposed to be the largest cell. The ostrich egg, it may be very broad. It may be up to 15. What we can say that is supposed to be 8 to 90 inch, or up to 15 centimeter. It will be greater in size. While talking about the two types of the cell, there are the prokaryotic cells and the eukaryotic cells. In prokaryotic cells, what is the difference between the prokaryotes and eukaryotes? The pro means the lack of the nucleus. Not only the lack of the nucleus, but the chromatin network. जैसे nucleus होता है, nucleus में क्या होता है? There will be a thread-like structure. Okay, that is a chromatin material. And what is the function of the chromatin material? To transform the genetic information. Within that chromatic material, there will be the chromosomes. What are the chromosomes? Chromosomes. Yes, chromosomes are supposed to be the vehicles of the heredity and variations. They can transfer the characters from the parents to the offspring. Okay, so we almost see just like our parents or sometime our brother or sister. So those characters, that hanging lobes and color of the eyes, okay, and the colors. So these characters can be transferred from the first generation to another, or from a parents to the offspring. That is the importance of that chromatin material, and that is a 20. How much chromosomes are existing in the human being? 23. That is 1 plus 22 means what is meant by 1? Yes, and 22 means that is autosomes and allosomes. Those can decide it can decide the characters of of the posture or the look. So those are very small. As I told you, those are not much greater. Their size is less than the 5 millimicron. It may be 0.1 to 0.3 millimicrons, that's prokaryotes, and different microorganisms and the blue-green algae. Okay, those are having a different characters. The absence of the membrane-bound organelles. What is happening? If you can see the prokaryotes. So so how you are observing? So this is a central portion of the cell or this organism or the bacteria or whatever you bacteria or the flagella. So this is the cell, and this is supposed to be the individual cell, and individual cell is having that flagellum. What is the function of the flagellum? Flagellum may help to migrate or to move the species from place to place. That is movement, that flagellum. And the chromatin material, it is not too much specialized as we see in the eukaryotic cells, where the nucleus is there, and within the nucleus there is a chromatin material is there, and again the nuclear is again double membrane, two membrane, and within that membrane there will be the nuclear pore, and from that nuclear pore to the cell cytoplasm there will be a continuous communication. So that is possible in a eukaryotic cell, but here no such a advancement we may observe. It is very simple, and the cell membrane is here, but there is no distinguished boundary between the nucleoplasm and the cell cytoplasm. Okay, it is a capsule. So this is a last outer membrane. We can see the capsule. Then ribosomes is there for the synthesis of the proteins or the DNA. That's all. Okay. Now, while talking about the next portion, that is supposed to be the nucleus is supposed to be the central portion. It is sometimes the nucleus may be centric. Sometimes in a plant, in a animal cells, we see the nucleus in the center of the cell, but in most of the plants, it is a hexocentric. What is a hexocentric? So the hexocentric means it is not exactly located in the center. For example, why it is not exactly, who is going to pressurizing the nucleus to shift away from the center? So this is supposed to be the animal cell. In animal cell, there will be the central region. Nucleus will be located in the central region. But the vacuoles, let us see, it is a central, but I am drawing the another figure. This is supposed to be the cell wall or the plant. This is a plant cell, and in plant cell, the vacuoles are supposed to be occupying 50 to 90% portion of the cell, and therefore the nucleus is going to shifting to the corner of the cell. Okay, and this is known as a hexocentric. This is a hexocentric and it is a centric. Okay, so that is the difference. Now we see the different kinds of the cell organelles. Here in this figure, you will see the almost all the cell organelles. This is a nucleus, and nucleus it is rightly starting from here, suppose, and this is a nucleus, but it is a three-dimensional figure. In this three-dimensional figure, those are the nuclear pore. As I told you, the genetic information may be communicated. Sometimes you will see such structures from the cell cytoplasm. If I am talking about the animal cell, there will be a continuous communication from the cell cytoplasms of a single cell to another cell. Therefore, we are feeling a heat, pressure, temperature, whatever. So that is the reason, because cells is having a certain channels or the pores through which the different kinds of the physicochemical conditions can be detected by our skin. Okay. Now this is supposed to be a most important region of the cell. It is a center, it is a driving force, whatever you may call it. And there are certain organelles you will see in this cell, but while talking about the nucleus is having a double membrane, and double membrane, along with the double membrane there will be a different pores. Okay, nuclear pore, and here is a nucleolus. Nucleolus is also important. Although the DNA is supposed to be situated in the nucleus, but the RNA start the synthesis with the help of the protein, the replication of the DNA. Okay, so this is supposed to be a double helical model which was invented by the Cratson. That is okay. So this can be done by, this is supposed to be the single strand. This single strand RNA synthesis can be done by the ribosomes, and which can exist in the nucleolus, not in the nucleus. While talking about the centrioles are supposed to be almost centrioles we cannot find in the plant cells. Those are only in the animal cells. Then coming to the lysosome is also important. So what is the function of the lysosome? Later on we will see. So this is supposed to be the cell, and cell is having a different kinds of the organelles. Lysosome followed by the ribosomes. Ribosomes may be synthesized on the RER. What is RER? Rough endoplasmic reticulum. What is the function of the rough endoplasmic reticulums? So we will see later on. So let us see, or just keep in your mind, the endoplasmic reticulum is supposed to be a carrier. It is going to transferring some information. Like with the Golgi complex or the Golgi bodies, the inventor of the Golgi bodies, Camillo Golgi, he has awarded the Nobel Prize for his discovery. That Golgi bodies is also known for the packaging. It is supposed to be the that Zomato, they comes and provide food material to us. That Golgi bodies is also packaging and distributing, mobilizing the food material from place to place. So coming to the cilia, which is supposed to be not very prominent in the developed organisms. For a lower organisms, for in prokaryotes, we can see that cilia and other locomotory organs. Cell membrane it is supposed to be the cell of the animal. It is not a plant cell. We see the cell membrane in the animal cells, whereas the cell wall, it is made by the cellulose, very tough in the plant cells. There is smooth endoplasmic followed by the rough endoplasmic. Although the rough endoplasmic playing an important role for the repairing of the cell, for making of the cell wall, okay, or the cell membrane. But the SER is also existing. What is SER? That is a smooth endoplasmic reticulum. It is connected to the lipid and the fats synthesis distribution. Coming to the another point, that is a nuclear pore. As I told you, that is a channel and the exchange to the information from the nucleoplasm to the cell cytoplasm. The nucleus is made by these 5 component. Okay, that is a nuclear pore, nucleus, nucleoplasm, and nuclear envelope. For that is 4 component are very important: nuclear membrane, nuclear pore, nucleolus, and a nuclear, that is about nuclear envelope. That is an outer membrane, the two-walled or double-walled. Now, the rough endoplasmic reticulum is already discussed. In mitochondria is supposed to be the powerhouse of the cell. There is one theory. ऐसा कहा जाता है कि पहले ये क्या हुआ था, इन प्रोटोजोइक एरा, जो हमारी पूरी हिस्ट्री है हमारे अर्थ की, वो कई एरास में डिवाइड हुई. डू यू नो that is in Mesozoic era, the Jurassic Park, like this. Those days, what was happening, the cell was also very primitive, and cell was supposed to be, one cell was eating another cell. That is engulfing theory. Okay, तो एक सेल ने दूसरे सेल को खा लिया, तो उसका बाकी जो पार्ट है वो डाइजेस्ट किया, बट सम पार्ट वाज नॉट डाइजेस्टेड. दैट अनडाइजेस्टेड पार्ट सपोज टू बी द माइटोकॉन्ड्रिया. द माइटोकॉन्ड्रिया इज सपोज टू बी द इंडिविजुअल एंटिटी, उसका खुद का एक अस्तित्व है. व्हाई? बिकॉज इन माइटोकॉन्ड्रिया देयर इज ए डीएनए. माइटोकॉन्ड्रिया and the plastids in the plant cells, those are having a special entity, special identity, because those are having their own DNA. That is powerhouse. And what is the function of the powerhouse? Let us take an example of the human sperm cells. So this human sperm cells may carry up to, suppose, the head, which may carry 500 mitochondria. So the energy is very important. In efficiency can be increased of the sperm cells to move in a genital organ. That mitochondria is supposed to be the powerhouse of the cell, and every cell may have the various number of the mitochondria. In the sperm cells, there may be the more mitochondria as compared to the other normal cells. Now, while talking about this nucleus, do you think that the nucleus is supposed to be existing all the eukaryotes? No, isn't it? There are certain cells, those are not having the nucleus. They are lacking the nucleus. In example, RBCs. RBCs are supposed to be the cells which do not have the nucleus. Why so far? Why this is happening with them? Because the RBCs are supposed to be migrating from a small or major, very, very small cells or the capillaries, so that it has to change its continuous shape, so that it is very simple. And in plant cells, the nucleus is not present. Sometimes in the plant cell, those cells are supposed to be the phylums. Those phylum, that is the sieving or the network of the transportation of the food, right, from the soil to the different branches of the plant. Those are also devoid of the nucleus. So try to keep such a typical concept in your mind, and the questions may arise from there. There are some cells, those are not, although in a developed organisms, but within that also some species some cells are not having the special cell organelles. Cytoplasm is also important. It is supposed to be a jelly substance, jelly material. It is an organic in nature, and where the different cell organelles are supposed to be the floating. Okay. While talking about the secretory vessels, that vesicles are supposed to be, these vesicles may, right from the Golgi bodies, these vesicles sometimes may be converted into the lysosomes. Okay. While talking about the lysosome, coming to the next cell organelle, we will discuss the lysosomes specially. So this is all about the eukaryotic cells. Eukaryotic cells are supposed to be the developed cells. There is a special, there is a distinguished features, or there is a distinguished boundary between the nucleus or the cell cytoplasm in eukaryotic cells. And there is a membrane bound cell organelles. Some cell organelles are supposed to be single membrane. Some cell organelles are the double membrane. Lysosome is supposed to be a single membrane, that membraned organelle, whereas the mitochondria is supposed to be a double membrane. Whereas see the different kind of the organelles. And so this nucleus is having either in the animals and the plants and fungi, these are the in the different developed eukaryotic cells, you will find this DNA and other cell organelles, whereas in the prokaryotic cells, very simple structure is there. So as I told you already about the nucleus is supposed to be the center. Okay, so it is controlling center of the cells and houses the cells genetic material. सेंटर के अंदर क्या है, न्यूक्लियस है. न्यूक्लियस के अंदर क्या है, जेनेटिक मटेरियल. इट इज हाउसिंग द जेनेटिक मटेरियल एज ए डीएनए, डीऑक्सीरिबोन्यूक्लिक एसिड, एंड इट कोऑर्डिनेट्स सेल एक्टिविटीज लाइक द ग्रोथ एंड रिप्रोडक्शन. सेल का जो डिवीजन होता है, सेल का जो रिप्रोडक्शन होता है, तो इस तरह से होता है उसका सेल का रिप्रोडक्शन. लेट अस सी, देयर आर द टू वेज ऑफ द सेल डिवाइड. Cell may be divided by in the two compartments. This is supposed to be the cell. The cell may be divided. This is supposed to be the nucleus. That is a karyokinesis and the cytokinesis. Karyokinesis means the division of the nucleus first, followed by the cytoplasm and so on. This is supposed to be the karyokinesis, followed by the cytokinesis. Yeah, after the prophase, metaphase, anaphase and telophase, this kind of the stages you will see. It is a coordinator, and different kinds of the synthesis, protein synthesis may be done. These are the functions of the nucleus to keep the safe and the managing enzymes in the cytoplasms, gene expressions, DNA replications, ribosomes assembly. You will see those RER and SER is coming from the nuclear pore. Okay, means the nucleus is responsible for the synthesis of those ribosomes or the ribosomes. Next is the ribosome assembly. Okay, dense regions inside the nucleus, there is produces a ribosomal RNA, that is a small RNA. It has to be written like this, small RNA, and which can initiate the cell division. Coming to this next cell organelle, that is endoplasmic reticulums, as either the smooth and the rough, both are shown in the same figure. This is a rough endoplasmic reticulum. This is a small dot like structures. Okay, already the small dotted structure is supposed to be indicating the ribosomes, and the smooth is not having that kind of the rough structure. Next is supposed to be the lysosome. Lysosome is also important cell organelle, and this cell organelle is also known as a suicidal bag. So why so far it is known as a suicidal bag? That is having a different kinds of the enzymes. And if he thought, or if it thought, if the nucleus thought that the cells are beyond the repairing, so the cell has to be destroyed, then it can destroy the cell. And sometimes it is having some positive the means and the roles that has to be discussed. I do not find any kind of the reference for this NDA examination, but for the lower classes or for lower grades, I found that the lysosome is widely explained by the state board as well as the CBSE board. The lysosome is playing important role for the that is in phagocytosis and the destroyer. It is also known as a destroyer or the suicidal bag. But another is most important role has also been done by this, is supposed to be going to bursting during the fusion of the female and males. What we can call it as either the reproductive structure and sperms and the eggs. The outermost eggs of the human female can be burst by the sperms, and that sperms may have higher concentration of the lysosomes. That outermost boundary of the head of the sperm may burst the eggs and it may fuses with the nucleus of this egg, and this regulates the hormonal cycles and control the embryonic fluids. Okay, that is a embryonic fluid, and this is also important. Okay, it is also known as a destroyer of the viruses and bacteria. Okay, lysosome ruptures and release the enzymes into the that is intentionally destroy the cells, and process known as a that is a programmed cell death of the suicide. Okay, wherever certain foreigners or the foreign particles, foreign material, or just like those are not suitable for our body, poisonous things which is supposed to be entering accidentally inside the body, it can be programmed, it can be destroyed with the program of the lysosome. So this is the structure of the lysosome, the outermost membrane. Okay, already this is a Golgi bodies, and Camilo Golgi has invented this Golgi body, and it is also the packer and mover. Okay, it is also distribute. So it is all about the mitochondria. So this is the last cell organelle which we are going to discussing. Either it is sometimes known as a powerhouse of the cell. Okay, and this is supposed to be double membrane matrix, that is folding section is known as a matrix, and outer membrane is ribosomes is also existing within this matrix. So it was all about the today's session of thank you. Thank you so much. So for the next what we can say, your unit test. Okay, so this topic will be, it is already declared, it is already declared, but this is not, I mean to say, it is not the entire cell